this graduate student in my lab, Malia Moore, who grew up here in Southern California in La Jolla as a lifeguard, swimming with Zostera, grabbing onto it and pulling it, and that's what you do when you surf. She started this project and wanted to know, like, can we actually find reasons why the Zostera is failing?
Hello, everyone. This is I'm Liachko from Phase Genomics, and you're listening to Uncultured, a pod that we started where we chat with scientists about cool research that has come out. Sometimes it is always interesting, amazing, sometimes deliciously chaotic. And part of it we highlight genomic tools, the kind of genomic tools that we release, like proximity ligation and Hi-C and things like that. But so today we are going underwater to explore the genetics of one of the most important marine plants you've probably never noticed, eelgrass. And I'm joined today by Todd Michael, research professor at the Salk Institute for biological studies. Todd and I have worked on a bunch of projects together over the years. His lab works on all sorts of cool plant questions. So everything from tiny little things like duckweeds to big things like wheat and other things, good old Arabidopsis, all sorts of things like that. But recently they published a paper in Nature Plants on a really interesting genomic thing that happens under the ocean. It's a hybridization between two different species of eelgrass. And so I'll just kind of let him talk about it a little bit. But eelgrass is very interesting because, and I know this, certainly not from just trolling ChatGPT, but it turns out it's the thing that I feel like when I look under on pictures, I go, that's the most seaweedy thing I can think of, and it's not actually a seaweed. It's actually a flowering plant, which when I read that, I was like, I don't know. It doesn't, when you say it, it doesn't sound so crazy, but it's like a flowering plant that lives underwater, and like spores and stuff are getting spread by water currents and things like that. So it's a surprisingly important thing that I think goes unnoticed, and it also has really interesting genomic sort of properties. So, Todd, thank you for joining us. Welcome. Tell us a little bit about what you do and this paper.
Well, thanks for having me. So this paper, I think, is really exciting in many ways because it sort of merges together a lot of the new stuff that we're able to do with the sequencing, with Hi-C. But the plant, as you said, is like amazing. So unless you go in the ocean, you probably have not come across eelgrass, otherwise known as seagrass. If you're from the coast, you probably see it quite a bit washing up on the shore. And if you're lucky enough, you can go swim with it in the ocean. But it's a foundational species. So it's found up and down the coast, all over the world, different species of Zostera or eelgrass. And it's responsible for storing large amounts of carbon in the sediment. And that's actually really important to ecosystems in the coastal range. One of the problems is that because we also like to live in the same place that the eelgrass likes to live, we like to build things around it. And that causes disruptions in the ecosystems. It also causes disruptions in the beds. Sometimes they're just completely removed. And so there's an issue with us losing those very important foundational species that are capturing the carbon, storing the carbon into the sediment, which is a very important aspect of the carbon cycling. But it also is providing really nice ecosystems for different organisms, and that's also very important.
Because it has rhizomes, right? That's why it has like actual roots and things like that, which the seaweeds of the world generally don't, right? They just kind of like hang out down there.
Yeah, in general, seaweeds are brown algae, so macroalgae, green algae. So, a lot of times people mistake the two, although here in Southern California, macrocystis, which is the giant kelp, and generally what we think of as seaweed, around here basically is massive and breaks off and is on the beach as well, but looks more like an algae or a macroalgae.
Big giant thing.
So the seagrass is also important for what you pointed out. It makes this rhizome, which allows it to clonally propagate. So a lot of times when you look at a seagrass meadow, it's all just a clonal meadow of the same seagrass that's growing clonally through this rhizome. And some people call Zostera the saltwater sugar cane, because basically this rhizome is just full of sugar that enables it to grow even when the light is low, which is one of the things that we were interested in, because right now there's a big push to restore a lot of these eelgrass beds, meadows, and there's about up to a 60% failure rate of those restoration activities. So this graduate student in my lab, Malia Moore, who grew up here in Southern California in La Jolla as a lifeguard, swimming with Zostera, grabbing onto it and pulling it. That's what you do when you surf.
For the sugar, sweet rhizomes. Yeah,
exactly. And she started this project and wanted to know, like, can we actually find reasons why the Zostera is failing? And while she was doing this, she found this bed that was restored in a local bay here. And she found that it wasn't just one species of Zostera. It was actually Zostera marina, which is our local one in the intertidal, more shallow areas of the coast. But she also found this hybrid between Zostera marina and Zostera pacifica. Now, Zostera pacifica is a deep water species that lives off the coast just a little bit, about 100 meters off the coast, in 50 feet to 100 feet of water. So it has a completely different ecology. In addition, it also looks different. It has thicker leaves. It still flowers and does all those fun things.
And so the idea is that they're clonally, right? So these rhizomes are basically root structures, for people who don't know, right? That grow under, they're like ginger, right? Where they just go huge and they spread, and like new shoots come out. And so you can get this like meadow that's basically clonal. Like, you can tell by my name, we're mushroom people. And so mushrooms are the same thing, right? You can have one individual mushroom that takes over this entire area, and it just pops mushrooms out all over the place. It's a little bit like that. But then they also flower underwater, which still is mind-blowing to me. And then what is it, pollen, spores, what is it like?
Same process.
And that's how they make these, that's how they hybridize, presumably.
So eelgrass is called a grass because it's technically a monocot. It's not a grass like rice is a grass. They're in two different families. However, it is very interesting. In Southern California, that is like the bottom of the range, but it also is in New Mexico a bit, and you get more annuals down there. And so basically it has two life cycles, which it can be annual, meaning it sprouts, grows up, flowers, and dies. So that version doesn't have a rhizome, but it also can be perennial, and it can grow for several years, for 20, 30, 40 years, and basically continue to make sprouts, and those will flower. So it can both flower and also grow clonally. And in Mexico, where the annual is found, they actually harvest the seed and eat it as grain. So, and this was popularized by a Spanish restaurateur or chef in New York City, who was actually making dishes out of Zostera grain, which is pretty crazy. So it's not only a foundational ecology species, but it's also a food.
Nice. Yes, I'll check the Little Mermaid documentary, see if the food is properly. That's super interesting. You'd have to collect a lot of it to like make enough to eat, I guess.
Yeah, but they grow these big meadows of it, and then just like a field of rice or a field of corn, right?
It's crazy. That's super interesting. I should have done more ChatGPT research.
So you might not have been able to pull this stuff up. I mean, Zostera is just full of all these really interesting little nuggets.
That's super cool. And so let's talk about like the genome. So the genome of these plants is not huge, right? As far as right? As far as plants go, like certainly like wheat and all these like terrestrial crops, right, tend to be gigantic. These guys are a little bit
Yep.
smaller size. But it's the complexities, the hybridization that's interesting, right? So like,
Yep.
you mentioned tools. When we started Phase like a decade ago, putting a genome for something, building a real like high quality chromosome genome for something was like super hard. Um, Anytime there was a hybrid, it was like you just have to be like, well, bye. I'll come back in 10 years when
That's right.
technology has evolved. And now it's much more straightforward. So tell me a little bit about how you built that genome and sort of what the challenges were and how you overcame them.
So when Malia found that this hybrid was co-located in shallow water with Zostera marina, we thought, okay, well, this is a good opportunity to go ahead and sequence those genomes, both the marina and then the hybrid. And then we also collected pacifica because we have permits to collect up and down the coast and deeper water and then shallow water. And we basically sequenced all three. And really, a year and a half ago, based on the technology, we would have struggled to have made that into a haplotype resolved hybrid. We probably would have been able to do a couple things to, pull out specific parts of the haplotypes, but because of the sequencing technology that really has developed, the long reads through PacBio or Oxford Nanopore, we were able to put together both haplotypes, and then we used Hi-C to scaffold them. And even though the genome is a modest size of 300 megabases, it still was, at the time, like when we did this almost 2 years ago now, it was like a big deal that we were able to actually separate these things, because ultimately we wanted to look at why the hybrid could grow better under that environmental condition. And to do that, we really needed to see what was going on in the different haplotypes, the marina haplotype and the pacifica haplotype of the hybrid. So it was really the power of having the haplotype resolved chromosome-scale, so scaffolded with Hi-C genome. And, the marina genome had been published by JGI several years back in 2015, and that was a really nice genome. And then they updated it with a chromosome resolved genome several years back, I think 2020-ish. And so we had a nice reference genome to work with, but then we augmented it with several genomes up and down the coast, and then this hybrid and the Pacifica genome. So it was really great to see the differences between them.
When we first started doing genome builds, like again, years and years ago, the goal was everybody wanted to start with the most inbred homozygous thing they possibly could, because any amount of variability or haplotype diversity or heterozygosity or anything would just make your genome it would just become impossible to assemble anything. And now I feel like it's the other way around. Is that
now we look for like the most diversity, like hybrid is like the best case scenario, really, because it's super easy now to tear those apart.
Now with the better long reads and also with the Hi-C data, right? Hi-C traps physical connections between the sequences that are touching each other, and so you could even back in those days you could phase haplotypes. But now I feel like dicaryons, haplotype, like hybrids, polyploids, all that stuff.
And the nice thing about this hybrid is that since we had the marina and the pacifica, even though they weren't parents per se, that actually gave us some ability to also separate them using trio binning. So that actually was quite powerful as well. So we applied all these different new techniques that we can with the, both with the Hi-C, with the long reads, and then also with the trio. It was really that all these things came together to allow us to get that kind of resolution.
Did you see when you guys put together these hybrids, did you see gene loss that at sort of an elevated rate? Like one of the things we used to always see when we did, I come from the fungal world, and there's a ton of hybridization, but then they hybridize and they immediately start losing genes. Did you guys see any of that, or are they two pretty complete genomes?
Well, this is sort of the interesting part is that the Pacifica genome and the marina genome, both in the hybrid and then separately, are very much alike. So we did not find a lot of gene loss. We actually didn't find a lot of structural changes. We didn't find a lot of translocations. We just like the
Is it
genomes.
evidence maybe that it's a recent event? Maybe like climate change drove them to do it or something like that?
Well, so in the case of our hybrid, it was actually a restoration project where I don't know why they let them do this, but the restoration actually planted Marina and Pacifica in the bay,
Oh,
and Pacifica died,
really?
and the hybrid actually maintained and
Because it has
augmented
better
itself.
sugar in the.
Yep.
Yeah,
So it
yeah,
was
yeah.
almost like they did the experiment for us,
Yep.
thankfully, and we were just following up and finding out why this hybrid, is doing so well. But the bigger question is they aren't very different. So why aren't they very different? So we did also sequence some marina up and down the coast, and we found that actually marina from Northern California did have some structural variation compared to the Southern California version. So I don't know. It sort of starts to beg the question, like, are these actually really different species? Obviously we know that they can cross. We don't know at this point whether they make viable seed.
Right.
Although we are collecting seed
I think there
now
was
to
at least
check if it's
one
viable.
seed.
Yeah, one viable seed. Although what we did find is that so this restoration event happened like 10 years ago, something like that.
Mm-hmm.
And we only found F1 hybrids. And we actually sequenced a sort of a swath through the meadow, which makes us think that it is maybe an infertile F1 hybrid. But
Yeah, or like
we
it's
don't.
short-lived or something, right? Maybe
Yeah.
that's why it's not accumulating SVs because it dies
Yep.
before it can happen or something like that. That's
Yep.
interesting. Yeah, it just like I think about, like just the dynamics. Like I feel like because it's underwater, all the like pollen and all the hybridization dynamics must be so different than what we're used to here, right? Plants above, up like terrestrial plants, wind blows, bugs carry stuff around,
Yep.
but somehow I imagine the buoyancy of the water will change all that by a lot. But I don't know. It's kind of a cool.
It is super fascinating to think of the differences. Like most grasses are wind pollinated, so and the
Mhm.
flower actually looks very similar to say rice inflorescence, but another
I feel
great
like there's a
question.
lot of dad jokes in here. Why did the something with the eelgrass bring a flower? Like I feel like I'm close. I'm close to some dad jokes.
You work on it.
Yeah, I will. We all have jobs to do. Yes. Cool. What else are you working on? So that was a super interesting kind of paper. Always very convenient when someone else does the experiment for you.
Yep.
You're like, oh, I didn't mess with the water ecosystem. Someone else did. I'll just sequence it. What else is cool? What's coming up?
Well, we're working on several pangenomes, cassava, sorghum, maize. So one of the really cool things, just on a high level, is now that we're able to get the resolution of many different genomes, we can start to see like the diversity in some of these species that we just haven't been able to access, by the short read resequencing or other. And so we're actually seeing some interesting structural changes across the genomes. Like in cassava, we're actually seeing a structural variation, a translocation that's shared across the chromosomes that actually might impact some really important traits. So I think that's going to be a general thing that we're finding, is that these translocation and structural variants across these big pangenomes actually are playing a role in some of these very important traits. And so we'll see a lot more of those. And as you probably noticed, lots of plant pangenomes have been coming out, which is creating a great amount of data for us to just traverse genomes in a new way.
Yeah.
So that's what I'm really excited about, is now with the rise of like the large language models, the foundational models, and then just the number of genomes that we can actually utilize from a computational perspective, we can start to ask questions that we just weren't even able to touch before.
So I think that's a super important point, right? Like in terms of just the general progression of the field, right? We used to do genomes, we used to do genes, right? Then we started doing genomes, and now it's pangenomes because now we have the ability to go beyond one and actually do populations and things like that. And so yeah, so it's been sort of a steady march, kind of towards higher levels of information and things like that.
And now we can, I mean, I'm hoping that we get to a point with retraining these foundational models like Evo2, you know, that we might be able to start to get into, you know, even beyond like general like grammar of plant genomes, which would be, you know, one level beyond the pangenome.
Totally.
So I think there's some really fun things happening.
Just going towards towards the singularity. You can just understand all of biology computationally. One day. Anyway, well, that's very cool. Well, Todd, thank you so much for joining us. So your paper's in Nature Plants, and I'm going to have your student will appear here. Your first author, Malia, will be here. You mentioned her. And thank you for joining us.
Thank you.
Again, it's always a pleasure to work with you guys. And anytime good science comes out, it's fun for us to highlight it. And and just kind of like how what we've made helps people do crazy genomics things.
Yep.
So one more thing before we go, we kind of like to do this because it's fun. What is one scientific guilty pleasure that you have? I'll give you an example, like pulling parafilm for no reason and stretching it, or giving your students conflicting advice to make them go crazy. What do you?
Well, you know, when I was thinking about like scientific guilty pleasure, unfortunately, it's maybe not as exciting, but maybe more revealing. I actually just like to sit and align my genomes and look for strange things in them, which
therapeutic, yes. Yeah.
It's almost like doing jigsaw puzzles for me.
Once I take over, you won't be able to do that anymore. I'm sorry.
That's true.
Cool. All right. Well, thank you so much. If you guys want to learn more, check out Todd's paper in Nature Plants. We'll link it in whatever comments wherever this is posted. Also, follow us, follow Phase Genomics, follow this this series of recordings and this Uncultured podcast, and smash that subscribe button. I think I have to say it now that I'm doing podcasts. So thanks everybody.